What's up, guys? We are going to carry on with notes, and I'll let you know how those should look. But we're going to start out with the principle of independent assortment after our basic Punnett squares. And so, in the principle of independent assortment, we've just learned how to do basic Punnett squares, the monohybrids. Mendel needed to answer one or two more questions, like when alleles are being segregated, does the segregation of one pair of alleles have any impact on another, or does a specific gene that determines if a pea plant is tall or short have any impact on seed color? And so Mendel ended up designing a second experiment to follow two different genes as they pass from parent to offspring, and this would be known as a two-factor or dihybrid cross. We won't actually do dihybrid crosses, but we'll talk through them a bit. And so a pea's two genes, one parent had peas that were round and yellow, and another parent had peas that were wrinkled and green, and the round and yellow traits were dominant. So Mendel crossed these true breeding plants, so round and yellow with wrinkled and green, and found that all F1 offspring had yellow round peas. So we then allowed other things to self-pollinate. And when we allowed the first generation to self-pollinate, we actually did produce things with completely recessive um, genes. So the production of the first generation meant that the alleles for seed shape had segregated independently for the alleles for seed color, meaning that one gene had no effect on alleles for another trait, known as independent assortment. The principle of independent assortment states that when gametes are formed, the alleles of a gene for one trait segregate independently of that for another trait. So think about your hair and your eye color. They don't move together. They are very independent. You might get blue eyes from your mom, but blonde hair from your dad. Having one does not directly mean you'll have another or another specific trait. So a summary of Mendel's principles, they form the basis of modern genetics. They include that the inheritance of traits is determined by individual units known as genes. The genes are passed from parent to offspring. Each gene has two or more forms called alleles. Some alleles are dominant, others are recessive. Each parent has two alleles for a particular trait, and they'll only pass one to their offspring when the alleles segregate. The alleles for one trait segregate independently of another. But there are some exceptions. There's some exceptions to this principle, as not all genes will show a pattern of dominance and recessiveness, which is very annoying. For some genes, there are more than two alleles, so many times traits are controlled by more than one gene, and we're going to discuss some of these weird exceptions. So gene expression is always the result of the interactions of genetic potential with the environment. So with specific flowers, a seedling may have the genetic capacity to be green, to flower, and to fruit, but it will never do those things if it's kept in the dark. A tree may never grow tall if the soil is poor and has no water or oxygen, carbon dioxide available, oxygen available. And in other words, the presence of the gene is not all that's required for an expression of a trait. The gene must be present along with proper environmental conditions, right? Humans, we will have gene expression. We'll have blonde hair, blue eyes, a specific weight or like body height pending our genes. We will not reach that if we're malnourished and don't have food or water. The phenotype of any organism is the result of interactions between genes and the environment. So we have this thing called incomplete dominance or non-dominance. And so all traits are not as clear-cut as dominant or recessive traits. Some genes do appear to blend together. For example, such as snapdragons or 4 o'clocks, a homozygous red flower crossed with a homozygous white flower will yield a heterozygous pink flower. And this is known as incomplete dominance or non-dominance. And so no allele is dominant or recessive. They actually will blend together. And as you'll see in the next couple of practice problems, since there's no recessive allele, only capital letters are used. For example, the red flower is two capital R's, a white flower is two capital W's, and the blending, the pink hybrid, would be capital RW. So let's practice. So what type of offspring might be produced by two pink flowering plants? Well, what are the genotypes of that plant? We just said they'd be RW. So I'll give you a moment I'll remove those, and I'll give you a moment to write those down. When you're ready, I'm going to challenge you to fill out your Punnett square and your genotypes. Then we'll talk in a moment. So if you've noticed, and if you filled out your Punnett square, you'll see that we have 1 out of 4 capital R, 2 out of 4 RW, and 1 out of 4 WW. That phenotype would be 1/4 being red, half being pink, and 1/4 being white. We also have this thing called co-dominance, where we have in humans 4 blood types are possible: A, B, AB, and O. And there are actually 3 alleles that will determine this blood type. So the alleles are IA, IB, and I, lowercase I. Alleles IA and IB are codominant. The allele lowercase I is recessive. So codominance is when both alleles are apparent in the phenotype of the heterozygous offspring. That's a really big idea. We're going to break it down for you and see some real examples of this. The cow on the right is known as a roan, Roan, and roan coloring is between like a brown and a white animal, and we see that quite often. It's also a pattern of co-dominance. So there are many possible types of genotypes for blood as follows. So take a minute to pause this slide and fill these out. The genotypes and the phenotypes are all listed. It will help you out when we have a couple more practice problems, both here in this packet and when you have homework tomorrow. So let's practice some codominance. So what offspring might be expected if one parent has type AB blood and the other parent is heterozygous for type A? Again, we're going to have to break down and remember what heterozygous means. So heterozygous means that both letters look different. So what's the genotype of the first parent? IA, IB. The second parent, IA, lowercase I. And you're going to just plop those in on your Punnett square. Again, take a moment to fill out your Punnett square and write down your genotypes. Pause the video, work through it, and then start the video again when you want to work through the phenotypes, and we'll talk through it together. So hopefully you've noted that there are 4 different genotypes in this problem, which means that we're definitely going to have some different phenotypes, but it's actually more interesting than you think. When we're looking at our phenotype, our offspring might be that there are 2 out of 4 that could be type A blood. We see that because we have 1 homozygous A and 1 heterozygous A. We also have 1 individual, 1 offspring that might be type AB, and 1 individual, 1 offspring that might be type B, heterozygous B, that is. We also have genes that have 2 or more alleles. They're said to be multiple allele. It means that there are 2 or more alleles for a trait. Best example for multiple alleles is the color coat in rabbits. So color coat is determined by a single gene with at least 4 different alleles. Think about this as hair too as well, right? Or eye color. So these 4 alleles demonstrate a dominance hierarchy in which some alleles are dominant over others. And the 4 alleles for coat color in rabbits are as follows. Now, I know this looks tricky. So the 4 alleles are as follows. Your 4 that you're looking for are in the little boxes, right? So C is a full colored, often called wild type or agouti. CH is a light gray or chinchilla. CH is albino with extremities or Himalayan. And then the lowercase c is albino. I've also listed in left to right and from top to bottom the possible genotypes and orders of dominance. So full color is the most dominant type. Then we have chinchilla, Himalayan, and albino. You are going to want to write out each of these little genotypes in that line because it will be helpful in the next couple of problems. Feel free to pause this and then we'll move on. So let's practice. What type of offspring would be produced by a full color rabbit that had the genotypes of C, CH, that was bred with a Himalayan rabbit that was CHC? So there is our wild type. There is our Himalayan rabbit. And we're going to make our Punnett square. Now we have our genotypes, and we can plop those in on the sides and the top of our Punnett square. We're then going to fill out this Punnett square as we would with every other Punnett square we've done. My apologies that it's C's because they're sometimes difficult to find or to write out in the appropriate manner. But we have uppercase C, C, H, C, C, H, C, H, uppercase C, lowercase c, 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 H, C. And so we're going to write those down in our genotype section. There's one of each. They're all very different. So for our phenotypes, this is where we're going to have to go back, look at our list of graphs. Anything with a capital C is going to be full color. So we have 2 capital C's here for the full color. 2 out of 4 are full color rabbits. And the next allele of dominance is CCH, and we have 2 of those. So 2 out of 4 are full color, and 2 out of 4 are chinchilla. We're actually not going to look at that too much moving forward, but it's an important thing to learn because if you ever take a basic biology class in college and moving forward, it will definitely be brought up. We also have something called polygenic inheritance, which is the determination of a characteristic is the result of an interaction of many genes. So some traits such as size, height, shape, weight, color, metabolic rate, behavior, susceptibility for disease are not determined by one pair of alleles, but rather the traits are the cumulative result of a combined effect of many different genes, and that's known as polygenic inheritance. So polygenic inheritance is a trait affected by a number of genes, or polygenes does not show a clear difference between groups of individuals, but we'll see like a tiny graduation of differences. And most of our normal human traits are polygenic. Again, hair color, eye color, weight, height, skin color, right? We aren't all 5'5", 5'6", 5'7". There's a lot of variation in there because of our genes. We also have some sex determination work that gets done in the genetics world. So human cells contain 23 pairs of chromosomes. There are 22 pairs of autosomes and 1 pair of sex chromosomes. We already learned that. So in males and females, all of the pairs of chromosomes are the same except 1 pair. So the 22 that are the same are called autosomes, and the one that's different is called the sex chromosomes. 1 pair differs. Again, we've talked about that, but the sex chromosomes actually differ in structure. So females in the sex chromosomes will have 2 large X's, and in males they'll have 1 X and 1 Y. So again, females have 2 X sex chromosomes, males have 1 X and 1 Y sex chromosome set. There's a nice little photo on the side here to show you what a sex chromosome set would look like in a male. So there are many genes found on the X chromosome, but the Y chromosome only appears to contain a few genes, and since the X and Y chromosomes determine sex of an individual, all genes found on them are said to be sex-linked. So more than 100 sex-linked genetic disorders have been associated with the X chromosome, and sex-linked traits include color blindness, hemophilia, and muscular dystrophy, and they're all caused by recessive alleles. Since males only have 1 copy of the X, they will automatically have the disorder if they inherit just 1 copy. 1 copy. So it could happen by either parent, and you'd have issues on that chromosome. Meanwhile, for females must inherit 2 copies of the allele, 1 on each of their X chromosomes. So in order for that trait to show up, both parents would have to have the genetic disorder. Therefore, sex-linked genetic disorders are much more common in males than females. More males are susceptible to color blindness than females. Same with hemophilia, muscular dystrophy, etc. So when working sex-linked problems, this is just like when I had you guys write some stuff down for the bunnies. Take a moment to write both of these panels down. So we have XN and X capital N. That's a normal fission female. The first column is color blindness. The second column is hemophilia. We'll use these phenotype and genotype standards as we move forward. we'll have to have that language working together. Take a moment, pause this article, and then move on. So we're going to actually practice on hemophilia. So we have a normal woman whose father had hemophilia marrying a normal man. So they want to know what the chance of hemophilia is in their children. So we first had to find out the genotype of the woman's father. We know that she's normal, but her father had hemophilia, which means that he had this split um, genotype where he was a recessive carrier. Now, the type of the woman, she'd be heterozygous for that disease, for hemophilia. So she has one capital H, lowercase h, and the man is normal, which means he just has one capital H on his Y. And now, if we plug in the woman and the man's genotypes, we can fill out our Punnett square. These Punnett squares will all remain 4 squares, so they remain fairly simple. We just write about that in our genotypes box, and then we work on our phenotypes. And so, if you wrote out the list of genotypes and their matching phenotypes from the previous slide, it'll be pretty easy. But we will have 2 completely normal females. 2 females, they're double X, and there's at least one capital H in there, which will be dominant. We'll have 1 normal male, so an XY chromosome set with a capital H, and 1 out of 4 hemophiliac could be male, or 1 out of 4 males could be hemophiliac, meaning that they did receive the recessive trait from the genetic line. That's the end of our slideshow, so let me go back 1 second. We'll talk through more of these. We'll practice them in class. So please come with any questions that you have. In addition, I'll have uploaded quite a few videos from YouTube, the Amoeba Sisters, and a couple other really great platforms that discuss and talk about Punnett squares and these different types of inheritance. Now, while we won't utilize them completely, or utilize all of them, I should say, in our coming dinosaur project, we will utilize incomplete dominance in terms of color of dinosaurs. So that one will be really important. The ones we will find most common are blood typing and like incomplete dominance. Again, please come with questions, circle anything, highlight anything you want questions on. We're going to work through a lot of these problems in class when we get in our next class session. So I look forward to doing that with all of you.